All right, guys. This is going to be a video covering the just last few slides here of uh, the heart lecture. We're going to talk now about what we call coronary circulation, which is what enables our heart to be fed with the nutrients that just about any other, I shouldn't say just about, but just any other tissue of the body needs, because we know our heart is made up of living cells. Living cells need nutrients like oxygen and glucose, and also produce wastes. So our heart needs blood just like any others. That's because the nutrients that are in our blood inside of our heart, in those four chambers that we've been talking about, are not able to diffuse quickly away from and out of the blood when that blood's in our chambers. So the cells are not able to get the nutrients in that case. Thankfully, we have our own network of blood vessels to feed the myocardium. We call this our coronary, our cardiac circulation. It involves blood vessels called the coronary arteries, the coronary capillaries, and then the coronary veins. Coronary arteries, we know now that arteries carry oxygenated blood away from the heart. These are the branches that come directly off of the aorta. Pretty much just right after that, blood goes into the aorta from our left ventricle. The two major coronary arteries are the left coronary artery that feeds the left side of our heart, and the right coronary artery that feeds the right side of the heart. As you can see, there are many branches coming off of those. We'll kind of get into more of those in our next lecture. But these are the blood vessels that supply our cardiac myocytes, the muscle cells, with the oxygen and nutrients they need. And blood is able to flow into these coronary arteries best when our heart is in its relaxed state, so between heartbeats. I already said, the first bullet there, right and left coronary arteries supply the right and left sides of the heart. Our heart also contains many anastomoses, which connect branches of a given coronary artery or extend between branches of different arteries, which enables our heart muscle to receive sufficient oxygen even if an artery is partially blocked. So basically, they kind of connect between two different arteries. So hopefully always ensure that our heart is receiving the blood flow that it needs. Fortunately, I say hopefully, because sometimes that doesn't take place, where a coronary artery may be blocked and it may not have blood flowing through it anymore, which is what's going to be best known as a heart attack, kind of more anatomically, physiologically known as a myocardial infarction, or MI for short, which if it lasts more than a few minutes can cause death of the heart muscle to result, which can be very dangerous, especially if it's taking place in this big one here, when that feeds our left ventricle. And as we know, the left ventricle is the largest and has the most cells, so it needs the biggest amount of blood flow. After passing through our arteries, that blood is now going to flow into capillaries, where the actual exchange of nutrients and waste is going to take place, just like capillaries anywhere else in our body, which we'll kind of talk more about in our next lecture. And then from the capillaries, blood is going to collect in our coronary veins, which drain that deoxygenated blood after the exchange occurred, most of which ends up dumping into the coronary sinus, which is actually on the back side of the heart. So that's kind of why it's kind of hard to show in this picture. It's an anterior view. Coronary sinus is the major coronary vein. It's actually a vascular sinus. We'll learn what that means in our next lecture. That drains blood into our right atrium, just like our superior vena cava and our inferior vena cava. And we talked about that earlier in the lecture, so that's not any brand new information to you guys. Here's kind of a cool picture of a cadaver heart, where you can see that big coronary sinus compared to the other cardiac veins. None of these pictures, I promise you, though, will be on the exam. It's really kind of hard to tell them apart when it's just red or when it's just blue. What we're going to do in our next lecture is look at these same arteries and veins, but we'll color them so that they kind of stand out from one another, along with a lot of other vessels. So we'll wait and talk about that next time. Okay, moving on, we're going to kind of move into a little bit of a different topic on what we call histology. which, if you think back to the beginning of the semester, we learned that histology refers to the study of tissues. So I want to talk about the cardiac muscle tissue anatomical on this page. Comparatively to our skeletal muscle fibers, cardiac muscle fibers are shorter in length. Remember, skeletal muscle fibers are long, cylindrical, run the entire length of the muscle. Aside from being less or shorter, they're also less circular in transverse section. So they kind of have a little bit of an oval shape to them. And a lot of them actually have a kind of stair-step appearance because they've got this kind of branching going on. You can kind of see that here in this illustration at the bottom. Typically, they're about 50 to 100 microns in length, having a diameter of about 14 microns. If you think back, we learned that skeletal muscle was multinucleated. Here we see that cardiac muscle is typically uninucleated, with the nucleus kind of centralized in the middle of the muscle fiber. Occasionally, we do find a cell that may have a second nucleus, but typically it's just one. Back in our lecture on skeletal muscle, when we kind of briefly introduced muscle tissue, we talked about structures that are unique to cardiac muscle, what we call intercalated discs, which are thickened bands formed by the sarcolemma, the plasma membrane, to help hold the cells to one another. Okay? Features of intercalated discs are these interdigitating folds, which I'm going to zoom in and show kind of how that zigzag appearance is in this picture, to show that this is a way that our cells kind of lock together. These folds match the folds of the next door neighbor cell and create a nice connection between the two. There's a lot of surface area, a lot of point of contact, and so it's a nice strong connection between the two, along with helping to increase that strength of connection between the cells are these structures called desmosomes, which help hold the myocytes together. To me, I always think of them like welds that hold pieces of metal together, enabling the cells to pull on one another without pulling apart. Something that's very, very important and is essential. Otherwise, our muscle within the heart will kind of just fall apart. Now, not necessarily having to do with pulling apart or anything like that, but there's one last thing I want to talk about that are associated with the intercalated discs, and that are these structures called gap junctions, which are open doorways essentially to connect one cell with another, which enables an action potential to spread from one cell directly into a neighboring cell, and then onto a neighboring cell, and then onto his neighbor, and then onto his neighbor, which is super, super important, and will kind of lead me into our last slide, because when cardiac muscle fibers get excited, they contract. And with this ability to spread action potential from cell to cell to cell, the cells all get excited at the same time, and then they contract in unison all at the same time, which is super important because it creates a very, very coordinated contraction, enabling blood to only go in a specific manner out of the heart. When we're talking about the ventricles, because they contract at the same time, or when the atria are contracting, they're pushing blood down in the ventricles. What we never want is, one side of the heart contracting, the other side not contracting. We want, both ventricles contracting at the same time, both atria contracting at the same time. We don't want all four chambers contracting at the same time either. That's not a good thing. So, very, very important coordination going on. We'll get into that a little bit more here in the next slide. A little bit more here on histology, though. First, you can see a lot of mitochondria within our muscle fibers. They're larger and more numerous than they were in skeletal muscle fibers, taking up about 25% of the cytosolic space in cardiac muscle versus only about 2% in skeletal muscle fibers. We see down at the bottom the same arrangement of thick and thin filaments with sarcomeres, I band, A band, M line, H zone, zone of overlap, all that wonderful stuff that we saw previously. Same thing is found in our cardiac muscle fiber. And We should know that by now because we've talked about it, and one of the features that we talked about with cardiac muscle, along with skeletal muscle, is that it's striated, and everything to do with the sarcomeres is why cardiac and skeletal muscles are striated, that striped appearance under a microscope. Other things, real quick, the T-tubes, are quite a bit bigger, but they're less abundant in cardiac muscle fibers and located close to the Z-discs. And then the SR, the sarcoplasmic reticulum, remember, stores the calcium. See, it's not as prominent as it is in our skeletal muscle. As far as the pictures go, don't worry about them. I'm not going to worry about you on those exam. Okay, and finally, getting back to anatomical parts of the entire organ, we're going to talk about what we call the cardiac conduction system. The system of essentially wiring of our heart that controls the generation of an action potential, but also the route and timing that it takes as it flows to our heart. The cardiac conduction system develops during fetal life when some specific cardiac myocytes, the muscle fibers, basically kind of change into something more like a neuron. They can create and then route action potentials instead of contracting like a muscle cell. They act more like a neuron. It all begins with a structure called the SA node, short for sinoatrial node, which is a patch of myocytes up in the right atrium under the epicardium near the superior vena cava. And it creates a signal that spreads not only through the atria, the right atrium, but also across through the interatrial septum into the left atrium. And it's able to do that because of those gap junctions that are found in the cells of both the right and left atrium. So these purple arrows represent the spreading of an action potential. The sinoatrial node is not connected directly to anything else, but some of those action potential signals will arrive at the second component of our cardiac conduction system, what we call the AV node. And AV node is down towards the tricuspid valve and at the lower end of the interatrial septum. Because of the inner fibrous inner skeleton that separates the atria and the ventricles, this is the only route in which an action potential can travel. So this is our second node. Do not confuse the SA node and the AV node. Your SA node is where the action potential originates, which is why its nickname is the pacemaker. But the AV node is where it goes as it's going to then travel down into your ventricles. From the AV node, the signal is going to then all be connected by essentially a wiring setup, beginning with this short singular band, what's called the AV bundle or atrioventricular bundle or bundle of His, which travels down from the AV node and then forks, forming the left and right bundle branches. The bundle branches travel down through the interventricular septum, and then coming off of those, traveling throughout the ventricular myocardium, both left and right ventricles, are these what we call Purkinje fibers. And following signals traveling through the Purkinje fibers, the ventricles contract. So what this enables is the signal to travel through the right and left atrium essentially at the same time, causing them to contract. There's a small delay that takes place once that signal arrives at the AV node before it then travels down through the ventricles. By that time, the atria have ejected their blood into the ventricles. Ventricles are filled up, atria relax, and then the ventricles contract and eject that blood out into the aorta or the pulmonary trunk, depending on which ventricle you're kind of focusing on. As far as the picture goes, make sure you are able to identify those 5 different things that are labeled there, because they are very, very important to know. Okay, that wraps us up, so I will turn it off and call it a day.